I'm here with Dr. Marshall Porterfield, distinguished professor at Purdue and former director at NASA. Welcome to Wilder Future.
Thanks, Tyler. I really am happy to be here and get to share some of my experiences with you.
There are so many places we could start. It's really gotten me thinking about if we're going to go to Mars, and it looks like that's what we're doing, we're going to actually have to live there. And it seems like a lot of the work that you're doing is specifically focused on the mechanics of regenerative circularity. And that's one of my favorite topics. So maybe we could just start there and talk about how actually living in Mars is going to be hard and how we're going to do it.
Yeah, I think I've always been attracted to or obsessed with the idea of the space program and space travel throughout my entire life. And I think when I was starting to enter university at that age, I really started thinking about what the future was going to bring, and for myself in the future, but also too for humanity, and also in the context of human exploration. I really feel like that is our ultimate challenge as a species, to overcome this limitation of living on this planet and being restricted to living here versus living more broadly in the solar system and moving out. I do believe that has to be our future for many reasons. One is that we've really depleted the planet. I mean, if you really look at where we're at today in terms of, recently there was a paper that came out that was the inventory of all the mammals on the planet. What's left? And it's really humans. The bulk of the biomass of the mammals on the planet Earth is humans and our food. And what's left in terms of wild animals are like 4 to 5% of the biomass, the mammalian biomass on the planet is wild animals. Mostly it's humans, our cows, pigs, our pets, rats are high up there. Those are so we've fundamentally changed the planet at the ecosystem level, and we continue to push it beyond breaking point. Perhaps we don't know for sure where we're really at in the context of the carrying capacity of the planet versus human presence right now. We know we're outstripping that. We need to learn to live within that, because that's really the challenge of what we need to be able to accomplish if we ever are going to have any kind of human activity beyond the planet. Even going to the moon and having an outpost there, if we were really trying to resupply it like a camping trip like we've been doing, it's impossible. It would literally be economically impossible. So the logistics of commodities really become the driver for human exploration. And that's really the driver for a lot of our history. I mean, even what's going on right now in Europe is about Russia trying to control farmland, the foundations for food production in the next century. That's really what that is about. So when you start thinking about things at that level and start understanding how the world's working around us and how these commodity systems are driving our social political framework, then you start to see and understand how the whole issue of human sustainability is so central to human civilization, and civilization being able to support expansion beyond the planet. It really is about understanding biology. The gaps right now in terms of deep space exploration and technology are completely biological. And they have been for the last 20 years. We've known that. NASA went a different direction about 20 years ago with their investments and cut most of the major foundational programs that were leading to habitation, bioregenerative habitation. And that's really where it kind of enter into this story is as a graduate student, I found out about the CELS program, the NASA Controlled Ecological Life Support programs have been going on since the 80s and 90s. These are foundational programs in the tradition that was really envisioned originally by Tsiolkovsky and his work in developing really the foundations for human spaceflight and spaceflight mechanics. He really developed all that in the late 1800s and envisioned bioregenerative life support being a foundational critical technology. And his writings and his work in terms of the technical engineering work is still the foundation for how they teach aerospace mechanics in Russia today. His foundational work literally led to Sputnik, right? So his influence really is immeasurable. in that way. And to really understand that, so last time I was at NASA Kennedy Space Center, they have a new headquarters building. When I visited there, in the lobby of the headquarters building, they have a huge display dedicated to Tsiolkovsky. So the recognition, the understanding of the recognition of his influence is really foundational. And if you look at that and you consider what they did at Kennedy, they had really the core of the Controlled Ecological Life Support Program. We invented vertical farming there. I mean, the first LEDs, the first advanced hydroponic systems, closed crop production researchers really went to that next level at Kennedy Space Center, and they really advanced those technologies. But that was really the programs that were cut in and for to pay for SLS. And the argument was, well, if we don't have a rocket to go there, then we don't need you. Well, if you don't have us, you don't have anything, you have no way to actually live there. So we have this conundrum now, and NASA's dealing with the political and policy fallout of the decisions they made 20 years ago, which led to the cut of the CELS program really at a critical time when we were really getting ready to move into integrated human testing. We were getting ready to build the Bioplex facility at Johnson Space Flight Center, where we were going to do integrative, basically bioastronautics, closed systems testing, and being able to prove out that these technologies can, support humans in the way that we're developing these systems. But all those programs were cut at that time. The Chinese immediately took those plans and invested in it, and that became the Beijing Lunar Palace, which they completed the first one-year trials in 2016. So they basically took the momentum of bioregenerative life support that was kind of carried through from Tsiolkovsky and through the Russian and Soviet programs, through that influence in the US and NASA programs, that when that was cut, the Chinese adopted that. And that's really the centerpiece for their International Lunar Research Station plans right now. It's also the core foundation of their space station program right now. If you look at the research they're doing, it's really focused on bioregenerative systems, crop production, food production. They've really taken it to another level beyond what NASA's been doing. And I can get into, what NASA did instead with the space station investment and how we kind of have wasted that over the last 10 years when we should have really been focusing on these human habitation challenges instead of the generic commercialization of low Earth orbit it's kind of sucked up all the funding, really.
It doesn't seem like there's a lot of primary research going on in space, as far as I understand. I don't know.
Things have been off track for a while in terms of the primary research we've been doing in space and why we're doing it. If we're using low Earth orbit to understand biological systems and fundamental science so we can develop technology to overcome those gaps in human spaceflight capabilities, protecting human health, protecting human productivity in low Earth orbit. Right now, we can't do that. Right now, when they send crew up to space, when they do all their training on the ground and they measure what your performance is, it's X on the ground. Whatever it is on the ground, they cut it in half in space. It's going to take you twice as much time to do the same procedures that you've trained over and over again in space. Why? Because of cognitive impairment and other critical human performance issues. And that's really what we've been trying to get at with the biophysical work that we've been doing and trying to understand the spaceflight environment from a biophysical perspective, because those are the kinds of problems we need to solve first in order to engineer hardware, atmosphere, spaceflight systems that are going to be at least equivalent to ground. So the perfect example, I saw last week that the Chinese were doing these candle demonstration experiments on their space station and putting out all this press, and it's really a fundamental experiment that was done actually I think they did some candle experiments on Skylab and then also on the shuttle, and then there's a whole combustion program where we look at the physics of combustion. But the candle flame in space is an ideal model. I'm a student of Faraday because he advanced the candle model in his Christmas lectures as it being the ideal system to study all these different physical parameters of science. So you can understand all these different scientific principles by studying the candle flame of oxidation and volatilization and thermodynamics of heat, how combustion is working, and how the flame, he actually dissected the whole flame. It has the shape that it does because it's a volatile plume, and that plume is shaped by gravity, by buoyancy-driven convection. And without that, the flame shuts down and burns as a sphere. And it actually will burn for a minute or two before it'll go out, because it's completely a diffusion-limited environment. So what's happening with that flame is that all the oxygen that goes in for the combustion has to go by diffusion. So instead of pulling in fresh air all the time, you're diffusion limited, the flame burns as a sphere and it just goes out. And what we found is that that's really what's happening in spaceflight for biological systems. If you don't design hardware to overcome the absence of gravity in terms of its primary thermodynamic influence in atmospheric and liquid systems, then you're already limiting your biological system. And that's really what the mistake we made. We built hardware systems that would work on the ground, and then we flew them in space, and then they didn't work because the media that we're embedded in changes. It's not the primary effects of gravity on our body that's the main issue. It's actually the effects of gravity on fluids and gases, and that influence of how that affects the interface of biological transport.
That really struck me when I was reading pieces of your work, that you could actually asphyxiate when you were breathing because that lack of flow. I mean, that was not something you would expect, but it's not something I would have expected.
Yeah, so essentially what our model shows is that theoretically, if you made a person stand still and stand there for an hour, on planet Earth, with buoyancy-driven convection around you, that you would get oxygen resupply and CO2 is pulled away. But without that, you end up being entrapped in this bubble. And you really would end up just like the candle flame, trapped in a sphere of depleted gases where your exhaled CO2 is building up, and then you would start to experience hypoxia in that bubble. And in theory, you would just asphyxiate in your own bubble, just like the candle flame would. Another model, or another theoretical consideration would be if you were in space and you encountered a water bubble. If you encountered a 6 foot tall water bubble and you're 6 foot tall and that water bubble is moving towards you, if you tried to push it away or made any little contact with it, that water would just go, you would be inside of it.
Shit.
Right? You would literally be trapped inside that bubble, and you would never be able to get out because it wants to stick. It's sticking to you and sticking to itself so strongly. That's one of the things that is so amazing about space, is that once you remove gravity, water has a completely different. So basically, if you had a pot of soil and you water it on the Earth, it drains out. But in space, the cohesion, the sponge, the matrix potential of that soil system would just pull the water in. So you end up with just a block of water. There's no air in the soil system. So root systems tend to respond to space as if they're hypoxic. And the most successful growth media we've used in space is essentially agar gel, which is just essentially a block of water. Because you get the same performance, basically on the ground. If the plants can already stand living in hypoxic conditions, the root system works in space, kind of. Our work shows that it doesn't. I mean, our original work in Chromex, we were looking at, my PhD advisor at the time was working on plant reproduction in space, and we flew the first series of experiments. We had Arabidopsis growing in these small chambers, and they were closed, but there was no airflow in the chambers. So when we flew those in space, the plants had flower buds. They were sitting there ready to flower. When they came back, they were almost like they didn't grow because they really couldn't absorb the CO2. They were in such a diffusion limited environment in those chambers without any airflow that they were like, the air was like a gel. So they really couldn't photosynthesize and grow normally. And then we did the next experiment and we introduced a small amount of airflow in the chambers and we get normal reproduction.
Wow.
So just And really the first experiment was kind of extreme because we had no airflow generation system in the chambers. So in space it became stagnant, and then on the ground, because of buoyancy driven convection, you still had just basic airflow. The plants could still grow enough in the chambers to reproduce on the ground. But in space, that the way we tuned the system, it failed. So we could see that biophysical limitation in reproduction. And I was studying the mitochondria and the root system, and I really became focused on mitochondrial physics and physiology because of the importance of the mitochondrial system in all biological systems. And if it was potentially being affected by the biophysical environment, what was potentially going to happen if we start to introduce the radiation issue. So that's really where we're at now. Our understanding from the bulk of the data now coming from Gene Lab, it's wonderful because we can basically go in and pull data from all these other experiments, humans, mice, nematodes, flies, and say, well, what's the same and what's different across all these species? We can actually use life itself as a filter to understand what's happening in space. And those results have shown repeatedly that there is mitochondrial dysfunction shows up as a major feature in spaceflight across all these different systems. In the human, every single organelle system, every organ system, the primary stress response looks like a mitochondrial driver. So we have the biophysical issues and radiation. And that's really the focus of what we're doing right now with the NASA LEAF mission, is using the plant systems to understand that additional radiation impact on stress in biological systems, and understanding that additional component that would go through mitochondria or chloroplast systems. Our main working hypothesis right now is that energetic organelles interacting with radiation can propagate reactive oxygen species production. So the organelles that are already highly active in terms of electron transport become stressed in the radiation environment. Interacting with radiation is hypercatalytic. When you already have reactive processes happening, you're already trying to inhibit reactive oxygen species production from the mitochondria. That's being perturbed by radiation and also potentially by the biophysical environment. So we think it's synergistic going through mitochondria or other types of energetic organelles. So we think the chloroplast is also going to be a major driver for radiation-induced damage.
That's a big deal because you're talking about the two arguably most important parts of the cell from an energetic perspective. So what kind of mitigation do you foresee?
So dealing with the biophysical environment, it's really about engineering the system to overcome the additional mechanical force you need to impart into the system so that you recover that normal biophysical environment. So even in this room I'm in right now, that light over there is a heat source, and it's creating all this convection and flow in the system. If that went away, I would literally be trapped in my own bubble here in this room. But it's not, because I have head space and I have a plume. Even when I'm sitting, my plume operates and it drives normal redox exchange. If that's being inhibited, we need to overcome that with better fans, better flow systems. We really need to see where the air is flowing and how it's flowing around the tissues and organs of the biological systems. I mean, this is no different than what we know that happens on the ground. If you have raising a bunch of plants in a greenhouse, a closed greenhouse, you can actually end up with CO2 being depleted, and the crop productivity will slow down. So if you look at greenhouse designs they have at the sidewalls, or the fans, and they're right at the canopy level. And the airflow through the canopy has to be at either around between 0.4 and 1 meters per second through the canopy. It's like a general rule for greenhouse designs. And that's really was overlooked. I mean, that, when we were moving these systems to space initially, that kind of engineering, really greenhouse engineering design in the hardware systems was not adequately done with our plant growth systems. And so if you look at the whole collection of Arabidopsis plant growth data from spaceflight for the last 10 years, a similar trend shows up. If you do a rack and stack it using Gene Lab and advanced genomics, what comes out is hardware. So hardware, the differences between different hardware systems that have been used for plant growth experiments with Arabidopsis are more relevant biologically than the difference between spaceflight and ground. because spaceflight and ground has been convoluted in these hardware artifacts.
What do you mean?
Well, if you have, just like the space station itself, when the crew members go up there, they aren't getting adequate ventilation because there's not enough airflow speed in the space station to break up those CO2 bubbles. So when you talk to the crew and you start to understand the astronaut experience, they're much more practical about dealing with these CO2 issues. Many of the crew members acknowledge and kind of operate, and so they're always trying to find, when they're working, they make sure that the fan is blowing air on their face. So think about it. If your body plume is a function of gravity, and that's based on buoyancy-driven convection, and because your body's at 37 degrees and the air is at 22. If the air gets warmer, you also can shut down your body plume just by increasing the heat, right? So how do you feel when you go into a warm room? You feel stuffy.
Tired. Yeah.
People always fan their face. They're trying to blow that CO2 bubble away from their face. They don't even know what they're doing. It's just subconscious. So what we really are seeing and learning from space now and understanding these CO2 and environmental issues is that these are actually the same problems we face here on Earth. We've closed ourselves into our homes, and with heating and cooling, that's sealed in. So we seal ourselves in with CO2. So most people are living in hypercapnic environments, and I think that a lot of our modern medical problems are the result of prolonged exposure to high CO2.
Wow. See, so this is one of the things that I notice in your work that I find fascinating is when we discover fundamental truths about how biology works that often, because we're looking at these edge cases and then we're looking at places like space where gravity has different effects, we have to really look at first principles. And that can often lead to innovations right here on Earth that you don't have to do because it's so easy here. It's, like we're in the womb here. It's hard to screw up. Whereas when we get into these more difficult environments, we actually have to know what's happening. We have to get it right.
Yeah, that's really one of the themes of what I feel like I've been doing in my career in pursuing this research to advance living systems into space is really we're understanding some of the most critical and fundamental aspects of life itself. And I think that's really where we're getting at in terms of our understanding of these organelle systems, especially focused on the mitochondria, because the central role it plays in human health. We know that the mitochondria is really the main controller for cancer. Apoptosis, which is the process for programmed cell death, will normally allow a cell that becomes damaged or precancerous to basically disassemble itself. And that process is completely controlled by the mitochondria. The release of cytochrome C oxidase from the mitochondria triggers apoptosis and will allow the cell to shut down. So two things have to happen with the cell for it to become cancerous. One of it has to lose control of its cell cycle, and the other has to lose control of apoptosis. So the mitochondria is the main controller for every type of cancer there is. We know that.
Yeah. Wow.
And it's the buildup of reactive oxygen species over a lifetime. What we're seeing right now and what we're understanding from spaceflight mitochondrial systems matches up really well with the emerging story about Parkinson's disease and how Parkinson's is associated with different agricultural communities where they've used rotenone, which is a mitochondrial toxin, and exposure to that has led to these Parkinson's pockets. They actually use rotenone now as a model for inducing Parkinson's in rat model for biomedical research.
So that's another thing that I'm curious about is when we look at our systems, they're incredibly fragile right now because there's this drive towards maximizing efficiency, and yet we have no clue what the consequences are, and they may be quite far down the line.
Yeah, I worry about what we will miss if we don't really slow down and pay attention to the biology and all the connections, because it's really not just understanding mechanistically how a biological system works. It has to be done in the context of ecosystems, because we don't really understand all the real connections between the microbiome, fungal systems, human biology, the nutritional elements that we get from a microbiome, that goes through fungal systems into food. Many of the nutraceuticals or vitamins that come from plants actually are not produced by those plants. They're actually produced by a microbial symbiont that's not completely known. The emerging story about ergothioneine in fungal systems, and which is also is considered to be a mitochondrial nutraceutical. These new discoveries really are emerging piecemeal too at the same time, but it's all stacking up in terms of understanding mitochondrial biophysics and the gaps. Really Mitchell's hypothesis is incomplete. That's really what we're discovering now from our work, is that Mitchell's hypothesis is incomplete because it only considers electrons and proton transport.
Can you
It's a
describe
quantum system,
Mitchell's
and
hypothesis?
there's light involved with mitochondrial physiology and biophysics. So it's really just like the chloroplast is an organelle that absorbs light, the mitochondria actually is producing light. And that light is a part of a quantum coherent feedback system. So we're starting to understand the biophysics of the mitochondria at a really extreme level now. That's really where I'm focused at in terms of those ultimately countermeasures. So one countermeasure is just shielding, getting away from the radiation. Microgravity can be countermeasures, would include biophysical transport. Now bone and muscle loading, those sorts of things, you're only going to get that if, with some kind of mechanical loading or artificial gravity. Ultimately, the best countermeasure for microgravity would be a centrifugal gravitational spacecraft. That would be the ultimate goal for that. On a lunar or planetary surface, you really can't create artificial gravity, but you can account for gravity in terms of airflow and liquid flow systems. Water flowing through pipes in microgravity sticks to the outside of the pipe. The pipes are actually hollow. The water's stuck on the outside. So everything flows and behaves differently in space. And most people can't get their mind around it unless they go live in space. But I've been obsessed with it over 30 years and trying to understand how it's impacting biological systems. And so we've developed a lot of, we have strategies for moving water, centrifuge systems that we're developing for biological experimentation. That's really one of the biggest gaps in terms of the ISS over the last 20 years, is that the original plans for the ISS included a large centrifuge module that was going to be provided by the Japanese. And then I don't know all the details of what happened, but the original plans versus what gets built and fly is usually different because of cost overruns and budgets. So it was cut. And the centrifuge module ended up in the parking lot in Tsukuba, Japan. But really, if you think about doing research in microgravity, if you don't have a 1G control in space that's getting the same spaceflight experience, spaceflight radiation, but a centrifuge where you get the 1G control to go with the microgravity, if you don't do that in space, then the radiation's not equivalent, and you never know what you're dealing with in terms of the radiation. And so that's what, if you look at the bulk of the research that comes through NASA, it's about spaceflight exposure, because they really can't discriminate between the microgravity component and the radiation component. So we're just massively ill-prepared.
That ties in so well with the patent you have on that centrifugal device. I was going to ask you about what's the purpose, because obviously it's interesting, but now I can see it's hard to actually accurately measure anything if you don't have that control, essentially.
Yeah, it's almost meaningless without that type of control. And the Chinese have actually been addressing this with their spaceflight research platforms in kind of an opposite way. They did a big experiment where they flew 300 mouse embryos. And this was actually the first mammalian embryo development experiment, large scale in space. They had a capsule system, so they flew the entire experiment in a capsule, microgravity exposure, measured the growth and development of the embryos. They saw effects in space. They measured the radiation during the experiment, and then they came back and they did a ground control where they applied the same radiation to the same experiment, and they only saw 1/2 of the results from space. So the other half, they said, well, that's microgravity. Well, how mechanistically is it microgravity? No one can't do, no one's done that full experiment. And it's really a simple experiment. I've written several proposals to DARPA and NASA to do that experiment. but they're just not really interested in it.
It seems so fundamental. I mean, if we're going to procreate moving forward, we've got to figure out, well, what's going wrong so we can account for that.
Yeah, it's really kind of a mess. I mean, in terms of some of the different research efforts that are going on in space and what the focus is and what the hypothesis is and the approach, you get a mixed bag. And so progress, again, has been slow because scientifically we don't have these kinds of controls that really hold people's feet to the fire in terms of existential impact and real first principles considerations for this work. Like you said, going back to first principles, that's really the level that I operate from. And a lot of research that's been brought into NASA would be for example, that there's a researcher over here who's been studying this certain type of blood cell his entire life. We think there might be something going on with those blood cells in space. And they'll just fly experiment after experiment and document all these changes, but without really getting mechanistically at how to solve it. So you end up kind of with these self-perpetuating, self-licking ice cream cone kind of research programs.
Yeah, where you're spending a bunch of money and time, but at the end you got nothing to show for it. Yeah,
you're producing all these publications, it looks like results, and ultimately you get an Alzheimer's drug that goes all the way into human, not beyond human trials, actually goes into the marketplace and completely fails.
It's missing that fundamental control.
Because it was just kind of a bubble that was floated forward. It was a research paradigm that had a bunch of publications and some high profile labs and lots of money going through there, and it looked like a lot of stuff was getting accomplished. But there was no real scientific advancements in terms of the real fundamental first principles problems.
Yeah, it seems like there's a lot of different areas that we need to get better at. One that I was thinking a lot about today, I was considering, your point about we're actually going to have to live there. There's so much to be said for human mental health and just systems actually working in a regenerative way over time, and closed systems, that I'm really curious about how we can set up more robust and just more of them test cases for small populations to be in closed areas for a certain amount of time.
Yeah, there's a lot of analog efforts right now outside of NASA, actually. There's almost like a little industry of analog astronaut trials, and it's almost like a tourist industry on its own right. So those are really important, putting people, we had the Mars 500 project that the Russians did. We didn't directly participate in that, but we had through the NASA Space Biomedical Research Institute, we were able to fund some USPIs to participate in that. But we also there's a 3D constructed kind of Mars habitat that they've been operating at Johnson, an Ohalo facility to test, some basic plant production, more like vitamin levels of plant production with humans in a closed system. And we know that there's great benefits in terms of human psychology, working and having plants. It's the same theme when you talk to people from McMurdo Station and Antarctica, where they've incorporated some of these systems to try to, as a test bed in those locations. They become very popular and really important to the crews that are there in terms of their mental health and socially. Just being around the plants becomes a central point for the research communities that are in those kind of locations. So we know it's going to be really important in that way too. But yeah, being able to essentially, what we really need to do now is move away from that human psychology component focused only and start to close these systems. And we need to have basically a true prototype where we put people inside of these systems and close them up and push them to the limit. I want to try to break it here. I want to build it and break it over and over again here so that we've solved all those problems. And we know that we can rely on that system to keep those people alive. There's people's lives going to be on the line on this.
Yeah, it seems insane not to test this really well. I mean, I was thinking about the Israeli Air Force, where they take people who fail a certain element of their checklist purposefully to actually check the checklist of what we need people to have in order to be effective pilots. But I was thinking about a similar context with these habitats, and for 3 months, give or take, you're going to be on a spacecraft. So you're going to be in pretty tight quarters. And then for 2 years after that, you're going to be building your habitat, and the whole time it's got to be regenerative. So I really am curious. I mean, how could you do this large scale? I mean, one thing I thought of was, what about students? You know, you could get credits, you could do education while you're doing it. You put them in a closed environment, you have 100 people per kind of habitat. And now you could start to really study a whole bunch of different parameters with these regenerative systems and start seeing what works and what doesn't.
Yeah, actually, I've been working with the student organization here at Purdue, Search Club, and they've spun off one of their main projects right now is basically a habitat program. And so many of those students have been working through MDRS, Mars Desert Research Station. We send 2 or 3 crews a year from Purdue, the Search Club. We typically have 1 or 2 crews a year, and I work with them on their research. We had a student who did a mycology project in the MDRS recently, but it's not closed, right? It's just you're going out there and it's a desert and you pretend like it's red and it looks like Mars. And I convince the students that you don't need to go all the way to Utah, pretend like you're on Mars. You can do it right here.
See, this is the thing that I've been
And the
thinking
important
about.
thing is not
Yeah.
putting on the spacesuit and going outside and pretending like you're picking up rocks. The important part is being closed.
Yes, exactly. And you can do that
challenge.
anywhere.
Yeah, so that's exactly what we're doing now here. We've got a freight farm container and a couple other trailers right now we're setting up to basically do some habitat trials. We're not going to have full closed system here. We're starting to collaborate also with Biosphere 2 and the SAM facility there. And so we're going to be bringing some of our systems there and testing them where they have closure of the atmosphere. We're going to be working on waste loop closure here at Purdue. Which is, starting with, you start with the first worst problem, poop. Because, that's anytime there's a natural disaster, the first problem you have is people get
Getting sick, E. coli.
infected.
Yeah.
So we've got to, and nothing will make you lose your appetite more than, no matter how good the food is coming up in space, and the crew food experience is so important, nothing will make you lose your appetite like smelling. So we need to solve that problem. And that's actually what we've been working with the group at Kennedy Space Center now. We've got a platform where we're using human waste streams to produce materials that we're actually using to make primary bioproduction capabilities. So we're actually making mycoponics. We're making the components for doing plant and fungal bioproduction from lunar regolith, where we're using waste stream materials as to create artificial binders. So we're making a bioceramics that we can print on the moon.
That's so cool. The other thing that I've always kind of considered in terms of closed loop systems is the difficulty of fossil fuels, getting them, making them, having them. And, really, your poop is several steps, but it's on the way to becoming fossil fuels. I mean, certainly you can make methane pretty easily out of it, and then you can have microbes that feed on that. And then I think that's a pretty interesting area as well. Really making ceramics sounds amazing. What other opportunities do you see in the recycling element of the bio waste?
Yeah, so the waste stream process that we're working with right now is actually being used by the group at Kennedy for producing a material called geomat. So they're actually producing that as basically a construction panel. And then what we figured out is we can use that foundation as a way to also make these ceramic materials. So they have a saying on the ISS where they have the water recycling system up there. They say, today's pee is tomorrow's coffee.
Right.
But on the moon, today's BM will be tomorrow's coffee cup. We're going to build hardware.
That's cool. That's really cool.
And not only are we building coffee cups with these ceramics, these are water filters. So the water filters themselves are some of the most costly commodity consumables that are used on the ISS right now today. Resupply of filters for the water system is still 80% of the up mass of just hauling the water up there.
Whoa. That's serious. I didn't realize it was that much. I mean, both from a volume, because you're essentially sending up a hard sponge, and the weight. I mean, the filters are a big deal.
Yeah, so being able to produce those kinds of capabilities and would be is game changing, really. So that's one of the things we're really working on now is really material systems,
So I'm curious,
biomaterials.
with the ceramics, you obviously need high heat. You obviously need some kind of base. I mean, ceramics are generally made out of some degree of clay or something like that. So maybe walk me through what you can about what something like that would even look like.
Well, we have normal kilns, and we would have a kiln, and we can produce bioclay using materials that are derived from a human waste stream that we're using as a binder. So once we use that as a binder, essentially we can make a clay body. And once you have a green clay body, you fire it. And once you fire it, it kind of melts in place, and then you get the solid ceramics. On the moon, there aren't natural clays, because natural clays are produced by weathering of minerals, which is really interesting because there are clays on Mars that you can recover. So we've been looking at using this platform as an alternative way to make basically a bioceramic, so we can make these ceramic materials in the lunar environment using the lunar mineral resources to do that. So this technology is going to allow us to basically create a new ceramics technology,
That's exciting.
which will
That's
spin
really
off and have applications here on Earth. And yeah, they're doing a lot with ceramic materials right now in terms of even replacing metal parts, machine metal parts. You can fire them and you get the same kinds of, some cases, better mechanical properties than some metal machine parts.
yeah. Some friction things I would imagine would be better with ceramics.
Yeah.
Yeah. Wow. Now with Leaf, you must be so excited about the Leaf program, and that's coming up. Do you want to tell people a little bit about that and what you were hoping to achieve with that?
So in the leaf experiment, essentially what we're doing is we're doing some of the first plant growth experiments in the lunar environment. We have a small modular plant growth system that'll contain seeds and starter materials for the experiment. It'll be actually a payload. It'll be launched with the HLS, the human landing ship, in that ship, and it'll be launched prior to the crew. So it'll be in place in the HLS around the moon when the Artemis crew comes and docks with the HLS, and they'll take that to the lunar surface. And then the payload will actually be taken out of the lander and put on the lunar surface on legs and activated with solar panels. And it'll run off solar energy on the lunar surface during that lunar day. Inside of the box, it provides lighting, humidity, atmospheric control for the plants during that plant growth experiment. We're going to be able to measure CO2, plant growth exchange with those kinds of parameters. But we also have a module that will basically undock and fix a portion of the plant growth experiment, and we'll be bringing that back and analyzing it with transcriptomics to look at gene expression patterns, looking at broad gene expression patterns associated with stress responses in duckweed, Brassica rapa, and also Arabidopsis. Arabidopsis is kind of like our universal research organism for plants. It's like the mouse for plants. Brassica is important because Brassica rape is where rapeseed comes from. So rape oil, and which is an important commodity, comes from that. And then duckweed actually has a lot of different potential as a nutrient source. It has a lot of advantages in this kind of environment too.
Which duckweed are you guys using?
We're not sure. We're actually looking at 3 different species right now because we're trying to determine what would give us the best heat tolerance, which you wouldn't think necessarily would come into play, but because of the experiment, because of the limitations on how we're getting the experiment to the moon, it has to sit in HLS potentially for several months. So actually have to be preloaded. So the seeds and all that, the dormant forms of the plant have to be tolerant to heat stress, and we're doing some testing on that right now.
Yeah, brilliant. So this is totally out of left field, but panspermia, the idea that life originated on Earth from somewhere else, does that resonate with you at all? Or what are your thoughts on that? I mean,
Yeah, because of the kind of central role that mitochondria and biological redox processes play in terms of living systems, I've really become more and more interested in a lot of primary research in terms of the origin of life, because I think what we're really seeing is that the most susceptible parts of life to space are those very elements of life that evolved first, that were the critical emergence elements of living energy systems. So maybe I'm playing my card. I'm maybe in the Nick Lane school of, although I don't give him credit for actually coming up with that, although he is a major voice in promoting that idea now, that energy systems really were the driver for emergence of life, not really information. So a lot of people get obsessed with living systems that have DNA, a code, the code of life that creates life. Well, maybe it's not the code of life that creates life. Maybe it's the energetics of life that created information. And those systems and the emergence of those systems are really, fundamental to the idea of emergence of life itself. So the early Earth and the conditions of the early Earth are very different than the way the Earth is now in terms of those types of environments. But if you really look at the chemistry and biochemistry of organic systems, non-living organic chemistry, there's nothing preventing the formation of most of the biological molecules that are important to living systems. And we've seen this over and over again from different samples from not asteroids, but comet tails, and now with the new materials they recovered from asteroids, we see they're rich in organic compounds, rich in carbon, organic compounds. In some cases, you might be able to just take that and feed it to fungi directly, right? It could be a substrate. I mean, there's hydrocarbon food just sitting there. Amino acids, nucleotides that form in space. And also water may not be so rare in space either, because if you think about the proton emissions from the sun, right? So the sun is already producing half of water everywhere. So anywhere there's oxygen, you could form water in the solar system. So water is probably not as rare as we could go to the moon, we could take minerals and basically cook them and extract the oxygen from that. And with the proton radiation from the sun, we could use that to make water to some extent.
Whoa. That's a game changer. That's a huge deal.
So, but all these pieces are out there. The pieces of life are in the universe. Pieces of life are already populating the planet before there was life. What catalyzed that transition to self-perpetuating entropic system? The energy profiles, energy coming into the system. Gravity is playing a role in terms of receiving the energy from the sun. Gravity itself is a form of energy transmitted through the mass of the planet. If you look at what's happening at the core versus the outer shell of the planet, the outer shell of the planet is being oxidized by all this radiation coming from the sun. So we already have an oxidative, reductive battery system. All we need is life to click in. And life did click in and kept the oxidation of the atmosphere going, such that there was fuel coming from the outer shell to keep the inner core running. So life on our planet, the actual electromagnetic shielding of the planet, is a product of life. And that's why Mars doesn't have life, because it didn't have enough biological activity. It couldn't spawn enough biological activity to fuel its core enough to where it could self-perpetuate and create an electromagnetic shield for life to really emerge. So if you look at the chemistry, just the organic chemistry of emergence of phospholipids and the structural elements of cells that would have just been like bubbles, there would have been, really I look at the early Earth in terms of the organic chemistry that was already there, the inputs of light, almost like the whole planet was like a living system before there were cells. That the Earth was actually like a living organic system, acellular, like a giant cell. And it was eventually where cellular systems have emerged with information, and that information conversion happened. I'm running out of time here. I'm sorry. I'm getting ready to jump into another meeting. If you want to, we can continue to talk about this later on. I'm sorry, but
No, that's great. That was really, really just a wonderful touch base on just some of your work and some of your ideas. I would love to have another one at some point, but thank you so much for taking the time. I really appreciate it. Thank you so much. Talk soon.
Thank you.
Bye.